Hello, everyone, and welcome to today's AAPS eChalk Talk brought to us by Sapient. The title of today's talk is On the Mark, Identifying Non-Invasive Biomarkers of Target Engagement. It is my pleasure to introduce to you our speaker, Dr. Mohit Jain. Dr. Jain is a physician scientist with nearly 20 years of expertise in physiology, biomedicine, engineering, computational biology, and mass spectrometry-based metabolomics. Prior to founding Sapient, he formed and was director of Jain Laboratory at the University of California, San Diego. There, he led a multidisciplinary research team of chemists, engineers, mathematicians, epidemiologists, and physicians to develop next-generation rapid liquid chromatography mass spectrometry systems to probe the non-genetic landscape of disease across population-scale human studies. Dr. Jain founded Sapient in 2021 as a spin-out of the Jain Laboratory to expand upon the mission of accelerating human discovery and drug development through the nexus of high-throughput analytical mass spec, computational biology, and population-scale clinical studies. Dr. Jain, the floor is yours.
Thank you so much, and it really is a pleasure to be here today. I just want to start and say thank you to the AAPS for organizing this Chalk Talk and for all of you for joining us here today. As mentioned, my name is Mohit Jain, and I'm here today on behalf of this team at Sapient to talk about our ongoing work to identify non-invasive biomarkers of target engagement. Now, even before we get there, I think it's important that we take a moment to step back and really understand why assessment of target engagement is absolutely so critical to the drug development process. As anyone who's engaged in the practice of drug development is well aware, it's a path that's fraught with challenge, and over 90% of drugs that enter into the developmental pipeline ultimately end up failing. This is a problem that our team at Sapient has spent many years thinking about. And there's a number of reasons for why we believe there's such a high failure rate, several of which we've had the pleasure of speaking at prior AAPS seminars. Retrospective analysis by major pharma companies on their internal drug development pipelines have suggested that upwards of 20% of clinical stage failure during drug development is due to failure of target engagement. This, of course, has enormous consequences in terms of cost, time, and ultimately patient safety. Now, ideally, markers of target engagement would be clear at the very early stages of the drug development process, where they can help guide patient selection, dosing, and ultimately go-no-go decisions before significant cost, time, and risk has actually occurred. So what can target engagement biomarkers really tell us? Now, when we think of target engagement biomarkers, we can really classify these into several categories. Direct target engagement biomarkers reveal the specific singular target that is engaged by the therapeutic, whether it be a small molecule, a biologic, a gene, or a cell therapy. Secondary or pharmacodynamic markers can be equally critical and reveal not only target engagement, but a secondary pharmacologic effect, which is essential to the overall therapeutic efficacy of the agent. In this way, both direct and secondary biomarkers of target engagement can help guide or validate hypotheses in vivo, particularly in humans. Now, while we truly believe that markers of target engagement should be essential to all drug development programs, they are particularly important when there's speed bumps along the developmental pipeline and a therapy fails to show efficacy or benefit. Under these conditions, a target engagement marker, both direct and or secondary, can be absolutely essential. Absent a marker or measurement of target engagement, it can be very difficult to ensure proper drug dosing or understand why a drug, in fact, did not exhibit its intended efficacy. Was the target, in fact, invalidated, or did the drug essentially fail to fully engage the target in vivo? Now, the challenge is that in evaluating target engagement, this is a process that becomes harder and harder as studies progress. There are a variety of tools and technologies and techniques that can be applied at the cellular or preclinical stages, but these wane and become much more complex once development and drugs enter into the clinical studies. Now, this is where small molecule biomarkers, including those of target engagement, can fill a very important and necessary gap. Small molecule biomarkers, given their physical size and chemical properties, can be produced intracellularly, whether it be within the normal tissue or diseased tissue, and then actively released from the intracellular space into the blood, where they can be captured and measured as non-invasive markers of local tissue physiology. These small molecule markers can read out both direct and secondary or pharmacodynamic target engagement, as we'll show you in several examples towards the end of this talk. These markers are highly conserved from preclinical to cellular studies all the way through humans, which allow us to discover at every stage and translate across the various stages during drug development. Now, Sapient really represents a discovery engine for small molecule biomarkers, including those biomarkers of target engagement. And we really operate on three integrated components. The first component of Sapient is our next generation mass spectrometry technologies that allow for measurement of thousands of small molecule biomarkers in humans at unprecedented speed. And I'll speak further about these bioanalytical approaches in just a moment. These technologies are embedded within a deep biocomputational program here at Sapient that allows for rapid analysis of complex multidimensional data, including integration of mass spectrometry data with genomics, clinical outcomes, and other features to identify key biomarkers that drive forward actionable insight. We also have a human biology database that encompasses hundreds of thousands of biological samples from around the world, which have already been analyzed using our mass spectrometry technologies. And in each of these biological samples, we have deep demographic, genomic, and clinical phenotyping, which allows us to amplify or leverage the underlying discovery process. Now, at the heart of our discovery operations is Sapient's bioanalytical technologies, which really represent next generation rapid liquid chromatography coupled to high resolution mass spectrometry. Here's an actual image of Sapient's high throughput laboratory that's based here in San Diego. Our tools allow us to utilize a variety of biosamples from blood to plasma to serum to urine to CSF, eye fluids, dozens of tissues and tumors, all the way through cells and media from a dish. And in these biosamples, we can measure greater than 11,000 small molecule biomarkers with four-dimensional chemical classification across a broad range of chemistries. We handle these analyses at unprecedented speed with an analytical cycle time of less than 1 minute per sample. And given a fully automated pipeline from freezers through sample handling, excisioning, processing, and mass spectrometry, we have capacity to handle tens to hundreds of thousands of biological samples at a given time. We have and believe in full data transparency and have in place rigorous quality metrics, and data can be integrated from external samples along with hundreds of thousands of biological samples previously analyzed at Sapient and embedded in our human biological database that I just mentioned. Now, in the setting of clinical trials or even preclinical studies, these approaches can be leveraged to identify new biomarkers of target engagement. In a very simple conceptual overview, biosamples can be acquired before or after exposure to a given therapeutic, again, whether it be a small molecule, large molecule, a gene or cell therapy. And samples can be analyzed using our mass spectrometry approaches, capturing and measuring thousands of small molecule biomarkers. Then, using biocomputational approaches, including regression statistics, all the way through much more advanced machine learning algorithms, we can identify those key biomarkers of target engagement. Alternatively, or in a very complementary manner, we can also leverage human genetics to identify new target engagement biomarker. Within Sapient's human biology database, we have tens of thousands of individuals in which we have dense genotypic information and matched blood samples with already measurement of circulating small molecule biomarkers. We can essentially integrate these two rich data sources to uncover new specific biomarkers that read out changes in gene function. These markers represent new direct and secondary markers of target engagement. So what does this actually look like? So if we look across the human genome, as we see here in this modified Manhattan plot, with genetic variation across all human genes shown on the x-axis, and the negative log p-value for association between that specific genetic variant and circulating biomarkers shown on the y-axis, what we see is that the higher the peak here, the more significant the association, starting from 10 to the minus 8 all the way up to 10 to the minus 200. As we can see from this global overview of the genome, there are thousands of small molecule biomarkers that associate with genetic variants at very statistically relevant thresholds. Now, if we focus on coding variants within the genome, there are naturally occurring genetic variants that affect specific gene function. And if we look across the entire human genome of over 20,000 genes, we've now identified circulating small molecule biomarkers for over 90% of all human genes. This includes 90% of FDA-approved drug targets, as well as over 90% of common genetic variants that have been implicated in human disease processes. So how do we take these approaches? How do we take these technologies and these data sets and use them in the setting of the real world to discover new target engagement biomarkers? And we have a couple examples here that we'd like to share with you. As mentioned earlier, lack of target engagement accounts for a very important fraction of overall drug failure. And here is one such example. This was a study from a phase 3 clinical trial of approximately 250 individuals that failed to reach its primary endpoint for a new therapeutic. Plasma samples were acquired from each of the study participants and subjected to our biomarker discovery pipeline, assaying in each of these biological samples thousands of circulating small molecule biomarkers. Each of these biomarkers is shown on a dot on this volcano plot here to the left. We can begin to statistically ask, are there markers that are only present in individuals that receive the drug versus a placebo? And when we do these very simple statistical analyses, we find that there's a single marker shown here in the upper right that's highlighted in red and circled in blue. And this single marker here is enriched only in individuals that receive the drug. Now, when we look at this marker specifically in individuals randomized to receive the drug, and look at the levels of this marker from baseline to post-treatment in the middle image here, we see that there's quite a bit of variability in the change in this target engagement biomarker post-treatment, with a portion of individuals having high levels relative to baseline of the target engagement biomarker, consistent with good target engagement, in other individuals having minimal to no change in this target engagement marker, suggestive of poor target engagement. Now, if you recall, this was a trial that failed to reach its primary endpoint for efficacy. And interestingly enough, if we stratify study participants according to those that had a good target engagement marker response versus a poor engagement response on the right here in this Kaplan-Meier plot, we see a very different end phenotype, whereby those individuals that had high elevation in the marker consistent with target engagement had a very, very different treatment response relative to those individuals that had low target engagement. This underscores the importance of evaluating target engagement as part of these clinical studies, particularly when a study fails to reach its primary endpoint. Now, here's another example in which we can leverage human genetics to identify really important biomarkers of target engagement. As many of you are well aware that work in the oncology space, the PD-1 ligand system has really emerged as a transformative therapy for solid tumor biology. And we know from clinical studies that there's significant heterogeneity in the overall response to drugs that target this biological pathway. To date, there really hasn't been a good marker of target engagement for these drugs. Leveraging our human biological database, we've begun to look for those small molecule biomarkers that associate with genetic variants in PD-1 and or PD-1 ligand. And you can see that in the top plot here, identifying a number of biomarkers that read out subtle changes in gene function among the PD-1 or PD-1 ligand area. We can take one such marker here, again, that's circled in blue, and we can cross-validate this in individuals receiving PD-1 therapy. And what we find is in that individuals that receive anti-PD-1 therapy relative to pre-treatment, this marker increases substantially with drug exposure, as shown here on the bottom left. As was the case with the prior example, we also find that in a phase 3 clinical trial for non-small cell lung cancer, those patients that have indication of greater target engagement, as measured by the single biomarker, had over a 50% increase in overall response rate relative to those individuals that had had lower levels of this target engagement biomarker. Again, highlighting the value of target engagement biomarkers in driving forward drug development and discovery. Now, I really wanted to make sure that we have enough time to go through questions, and I've hoped that I just presented here an overview for Sapient's approaches to target engagement, and we'd be happy to take some audience questions. Thank you again all for joining us. Here on this slide is our contact information, and you can email us at discover@sapient.bio. Our website is also listed here on the lower left at www.sapient.bio. And we'd be happy to stop here and time allotting. We'd be happy to take some questions. So thank you very much.
Thank you so much, Dr. Jain. We do have a bunch of questions that have come in.
Great.
Let's start with this. How do you envision using these target engagement biomarkers used in the drug development process?
Yeah, absolutely. So it's a good question. So, personally, target engagement should be a fundamental part of any drug development program. And I think most people would agree with that. The challenge, of course, has been that there's not been, for many, many therapeutics, again, particularly in the large molecule and/or gene and cell therapy space, there hasn't been good methods for deriving these target engagement biomarkers. Ideally, these types of target engagement biomarkers would be discovered at a very early stage in the preclinical scenario and confirmed early in phase 1, and would drive that development process all the way through phase 3, phase 4 studies, as we've given some examples here. That being said, there's a number of programs in which Sapient has engaged, in which we're putting the cart a little bit before the horse and starting at a phase 3, particularly for those assets that have failed to reach efficacy endpoints, and in using these target engagement markers, have attempted to rescue these agents through more target engagement work. So these target engagement biomarkers really can be applied and influenced at any stage in the drug development process.
Great, thank you.
Yeah.
Once a biomarker is identified, do you develop the method to quantify it in a GXP environment?
Yeah, it's a great question. So here at Sapient, as you can imagine, these high throughput systems are operating under an R&D designation. And typically there's a whole pipeline by which we validate these observations using canonical rLC-MS approaches, as well as have a high complexity laboratory here at Sapient that develops targeted and very quantitative assays. The importance of this is not only can we discover these biomarkers, but we can then develop the CLIA-based assay that allows for their implementation as part of clinical studies as part of that seamless pipeline. So oftentimes that is in fact the case by which we're measuring these thousands of things under an R&D designation, identifying the key markers, and then developing a quantitative validating assay.
Great, thank you. As you showed in an earlier AAPS talk, genetic modulate a small portion of circulating small molecule biomarkers, and these biomarkers are also under the control of environmental, diet, microbiome, and lifestyle factors. How do you then think about these target engagement biomarkers?
Absolutely. So it's really a very good and insightful question. As we showed you here, a number of these circulating biomarkers are under genetic influence, but they also are under influence of other factors. Again, whether they be environmental, diet, microbial, lifestyle, et cetera, et cetera. So one such classic example would be LDL cholesterol, which is a molecule obviously that's produced endogenously and is under genetic control, particularly within factors within the liver. But also can be influenced by dietary and lifestyle factors. Now, even if a factor or small molecule biomarker is under multiple regulation, if you will, by not only genetic, but also lifestyle or dietary factors, it still can represent a very good marker of target engagement. And again, going back to LDL cholesterol, this is a quintessential example. Again, a marker that's under control by many different factors, but really represents an exceptional pharmacodynamic and engagement marker for statin therapeutics, for example. So the fact that these biomarkers may be influenced by a variety of factors in no way invalidates, but rather enhances their use for target engagement.
Great, thank you. What if the small molecule identified for target engagement is not related to the mechanism of action of the drug under development?
Yeah, that's a really, really good question. And this goes back to the secondary or pharmacodynamic markers. And this is where we can begin to probe this using human genetics. So as you can imagine, there may be a number of markers that may emerge in blood in response to a particular therapeutic. Some of those may come from direct engagement and the mechanism by which that engagement occurs between the therapeutic and the target. And a number of these markers may have nothing to do with that direct engagement, but rather may be secondary markers that reveal downstream biology. Now, again, both of these can be critical in evaluating a go, no go, or validating a hypothesis regarding a particular therapeutic. And again, using human genetics, we can begin to understand whether or not the marker is directly related to engagement of that specific target or actually represents secondary markers. Now, as you can imagine, both of these have sort of orthogonal or complementary information. Whereas a direct engagement marker can tell us quite a bit about how an actual therapeutic is attaching to or inhibiting a specific target. Secondary or pharmacodynamic markers can read out the downstream effects of that. And both of these can be quite important in evaluating overall efficacy of a particular therapeutic. You can imagine a scenario by which a drug may be able to engage its marker, but due to underlying disease biology, the downstream pharmacology may not propagate in the correct manner. And so having markers both of direct target engagement as well as secondary pharmacodynamic markers, all of which may be revealed by these approaches, can be quite critical in understanding a particular drug and how it works.
Thank you. Have you given any thought to using target engagement biomarkers as means to retrospectively go back and evaluate therapeutics that have failed at phase 2 or 3 as a means of reviving these therapies and targets?
Yeah, it's a really good question and an area that we spend quite a bit of time thinking about. There's a lot that can be learned by failure, particularly in the drug development space. And as I showed you that one example from that failed phase 3 clinical study, one of the main reasons why drugs fail under these clinical scenarios is due to lack of target engagement, whether this has to do with dosing, drug-drug interactions, improper penetrance at the level of the tissue. Ultimately, what's happening is the drug is not engaging the target where it needs to engage it. And you can imagine that this type of discovery and target engagement biomarkers can help answer that underlying question. Was the target once again invalid, or was it simply the fact that the target wasn't properly engaged? In the case of that one example that I showed you, while this was a failed phase 3 trial that did not reach its primary endpoint, there's clear evidence that this was actually due to improper target engagement. Now, this can be used in a variety of different ways. You can imagine that this type of information can be used to stratify patients, and only those patients that demonstrate engagement of the proper marker are the ones that would continue along the therapy in a revised clinical study. Or it allows for altering dosing to try to understand how we can mitigate these target engagement markers in a way that allow us to revive a failed 3 asset in a way that we can show greater efficacy. So this is an area that's obviously of great importance to us at Sapient and to the field as a whole. And I suspect as we begin to deploy these approaches and these target engagement markers more broadly across drug development, this is going to be one of the real major wins by which there's a number of assets that have failed at the phase 2, phase 3 level that will be able to be revived through simply having a good biomarker of target engagement.
Great, thank you. Can you speak to how small molecule target engagement markers relate to DNA and protein biomarkers?
Yeah, absolutely. So when we think of biomarkers in human biology, oftentimes we focus naturally on DNA and protein markers. And these have been absolutely instrumental across a variety of human diseases and therapeutics. I think with small molecule biomarkers, these fill a very, very important role. Again, going back to the physiochemical properties of small molecules, these can be produced both in normal tissue as well as in diseased tissue. And given the size and chemistry, they're actively extruded from the intracellular space into the extracellular space where they can be measured in blood. Now, with DNA and proteins, it's a very different story. Only a small portion of the underlying human proteome is actively released into the blood where they can be measured. Now, these are very important circulating protein biomarkers, but these represent only a minority fraction of total proteins. And typically, until there's actual tissue necrosis or apoptosis of the cell, you don't release proteins into the blood. Similarly with DNA, as in the case of oncology and oncologic biomarkers, these have proven to be extremely important, but these are not really non-invasive markers, and they require sampling of the underlying tumor, which you can do in oncology, but for many other diseases, this becomes quite limiting. Again, this is where I think small molecule biomarkers can be extremely revealing. Because they truly are non-invasive, we can capture these in blood in the analogous way of a liquid biopsy, and allows us to tell us quite a bit of information about what's happening at the level of the tissue. So the way we view these is very much as complementary to traditional DNA and protein markers. And as you've seen from the examples we showed today, oftentimes there's much to be learned by integrating information from many different data sources, as we have here, in deriving these target engagement markers.
Great, thank you. How do you validate a target engagement biomarker in the sense of establishing a quantitative relationship to a pharmacodynamic response?
Yeah, so it's a really good question. There's a couple different ways in which we can go about doing this. In early phase 1 studies, particularly with dose escalation, typically are looking for a marker that's escalating with dose and related to an underlying outcome of interest, whether it be tumor regression or in other diseases, fibrotic regression, et cetera, depending upon the underlying disease. Oftentimes, as we discover these target engagement markers in human samples, particularly in clinical studies, we can go backwards, given the fact that these are highly conserved in preclinical and cellular systems, and ask, how does this target engagement marker operate and function in a preclinical scenario, again, where we can more sort of exquisitely assay underlying physiologic effects. And so in this way, we can not only establish the marker from human studies, but we can begin to understand its quantitative relation to long-term outcomes. The other way we do this is with our human biological database. As mentioned, we have these markers assayed in hundreds of thousands of people with dense genomic information and clinical outcomes data. So in the same way, we can ask within a contained phase 2, phase 3 clinical sampling, how does this marker act in response to a particular outcome? We can cross-validate that then in over 100,000 individuals using our biological database. And in this way, we can begin to understand the actual dose relationship between a marker and a particular outcome over time in very large populations.
Great. It looks like we have time for 1 or 2 more questions. Could you please elaborate more about primary and secondary target engagement biomarkers and give some examples?
Yeah, sure, absolutely. And I realize this becomes largely semantics and it's somewhat nuanced. So when we think of primary or direct markers of target engagement, you're really trying to identify a marker that reveals the engagement between a particular target and a particular therapeutic. That one-to-one relationship, when that marker, or when that drug, whether it be a large molecule, small molecule, gene or cell therapy, again, engages a target it was indicated to engage, is there a marker that's released that we can capture? And that's a direct marker of target engagement. Oftentimes when we see that marker emerge, we can validate it once again, looking at genetic variants that occur in that natural target. Now, there are also secondary markers. You can imagine that as a drug engages a target, there's a number of downstream physiologic effects that occur, pharmacodynamic effects, and that results in turning on of different small molecule pathways, and those secondary molecules may also be released into blood where we can measure them. Now, these provide orthogonal or complementary information once again. You can imagine that direct markers tell us if the drug actually engages the target, and secondary or pharmacodynamic markers tell us if it's had the desired downstream effects on the underlying biology. Now, this becomes really important once again in evaluating dosing and efficacy and outcomes related to a particular therapeutic. A therapeutic could theoretically engage its target but not have the desired downstream effect, or may not engage its target at all. And under both scenarios, when we have direct as well as secondary or pharmacodynamic markers, we can begin to tease that out in these human studies.
Terrific. We have one last question.
Sure.
Not sure if you've already touched on this, but theoretically, can you also use this approach to map potential secondary targets that may be engaged by a given therapeutic?
Yeah, so that's a really, really good question, and an area that we spend quite a bit of time thinking about. Because from our human genetic studies here, we've identified circulating markers that read out engagement for over 90% of the genes in the human genome. This provides some very interesting opportunities by which we can take an agent that has an unknown mechanism of action, or one that's believed to be heterogeneous in that it engages multiple targets. And after exposure to that particular therapeutic, we can ask what molecules are changing in the blood. Now we can begin to map those molecules back to many different genetic targets. And in doing so, you can retrospectively redesign, if you will, the various targets that are being engaged by these therapeutics. Now, this becomes extremely important given the fact that for many drugs, particularly in the small molecule space, these are believed to engage multiple targets. Rarely is there a drug that's only engaging a singular target, and that singular target is completely responsible for the physiologic effect of that therapeutic. And so we have systems in place here where we're beginning to deconvolute all the secondary targets for a therapeutic based upon these secondary markers that are released into blood. So it's an area that's rapidly emerging, if you will, here at Sapient, and an area that we're particularly excited about.
Well, thank you so much. That was our last question. Dr. Jain, any closing remarks you wanted to give?
Really, it's a pleasure. Thank you so much to everyone from joining today. We had really wonderful attendance, and we'd be happy to speak with anyone who may be interested in these types of discovery operations. So please feel free to reach out to us.
Thank you so much for a great presentation. And I wanted to let all of our audience know that you can find this recording and Dr. Jain's previous presentations by visiting the on-demand section of the AAPS website. Thank you so much, Dr. Jain. We look forward to seeing you in a future presentation.
Thank you. Have a great day, everybody.
You too.